Hello everyone, hope you are doing fine. So today's topic is about virus. Virus, the word which is came from Latin word, it means venom or poison. And viruses are so small, obligate, intracellular particles that is mostly can be seen only with the help of the electron microscope because it's so small and they must infect and take over to replicate. This is because they lack a chemical machinery for generating energy and synthesizing large molecules. And virus therefore must find an appropriate host cell in which they can replicate and as a result often can cause the disease. So viruses have some unique features not seen with any other living microbes to do all these activities. So they have no organelles, no cell wall, no cytoplasm, and no cell nucleus or nucleoid. Instead, they are comprised of two basic components, only two components: an indispensable nucleic acid core and a surrounding coat of protein. That is why a scientist who's called Peter Medawar remarked, a virus is just a piece of bad news which is wrapped up in the protein. So virus, when we say about the structure of the virus, you can see here a virus structure, very simple structure, surface which is having surface proteins, nucleic acid, and lipid layer and membrane. So the viral genome is very simple. As we said before, the viral genome of almost all viruses contains RNA or DNA. It can be either single stranded or double stranded. The protein coat of virus particle is called capsid, and that capsid gives shape to the virus. Generally, the capsid is subdivided into the individual protein subunits called capsomere, and capsid with its enclosed genome is referred to nucleocapsid. You can see it there. A capsid with its enclosed genome is altogether called nucleocapsid. And this capsid also protects the viral genome against the chemical and physical agents and other environmental fluctuations. And the viruses is also composed solely of nucleocapsid. It is also referred to as non-enveloped or naked viruses. And in some other type of viruses, either a special capsid protein called spikes or receptor binding proteins protrude from the surface, helping attach the virus to the host cell and facilitating its entry into the cell. And the nucleocapsid of many other viruses are surrounded by a flexible membrane known as envelope. Enveloped virus also contain a layer of two of the protein between the capsid and envelope called matrix that holds nucleocapsid to the envelope. A completely assembled and inert infectious virus particle outside its host cell is known as virion or virion. Okay, this is the basics about the structures of the virus. So you can see here the spikes there, which is protruding out from the surface, and there is nucleic acid, and surrounding it there is a nucleocapsid, and then lipid layer. Okay, and this structure or this virus is retrovirus, the virus of HIV, which causes HIV. It is really complex. It is having these spikes GP41, GP120, and it's having an RNA genome. Nucleocapsid is there and reverse transcriptase enzyme is there. So this is the structure of retrovirus. So as we said before, the virus is small in size and can be acellular or non-cellular. They are nucleoproteins and capable of multiplication only in living organisms. Mode of replication is very unique, and synthesis of virus-specific substances take place using biochemical machineries of the host. And here on this history part, there are some vaccines against the virus-caused diseases. In 1796, Edward Jenner found found out the smallpox vaccination. And in 1885, Louis Pasteur found rabies vaccine, and Charles Chamberlain found rabies agent is infiltrable. And 1892, Dmitry Ivanovsky mosaic disease he found out the mosaic disease in tobacco is filterable. So some of the findings which is related to the virus is mentioned here. And the unique features when we say about this in a nutshell, the virus is obligatory parasites, non-cellular, biological entities having DNA, RNA as genetic material, able to reproduce within the host cell by assembling their components, synthesized by using the biochemical machineries of the host. As we said before, we can see this viruses is so small, and we can see it only with the help of electron microscope, and they must inject and take over to replicate, because it lacks the chemical machinery for generating energy or synthesizing large molecule for itself. So it must take over some other host cells. So that is one of the unique features of the virus. And there are two types of life cycle seen in viruses: lytic cycle or lysogenic cycle. So when we get into this, the lytic cycle involves the reproduction of viruses using a host cell to manufacture more viruses. The viruses then burst out of the cell. The lysogenic cycle involves the incorporation of the viral genome into the host cell genome, infecting it from within. So these are the two types of life cycle seen in the viruses. And then before it, I want to say about the term bacteriophage. Bacteriophage is the virus which infect the bacteria, and that virus is a really complex virus, and they can follow two cycle, lytic or lysogenic cycle. And lytic cycle is followed, the lytic cycle following virus is called virulent phage, and those which can undergo both of them is temperate phage. So here we can see the diagram, lytic cycle and lysogenic cycle here. So when we say about this lytic cycle, first the bacteriophage. When we say about the bacteriophage, it have a head portion, collar portion, and the tail portion to attach to this host. The bacteriophage, and it is having a single nucleic acid on its head, and this is injected in a bacterial host cell, and bacterial host cell have a bacterial DNA, as you can see. So that first step is called attachment stage or adsorption stage, and that attachment is mainly by the chance collision. That is a result of some weak bonds. The tail fibers of this bacteriophage will bind onto the cell. So this is the attachment. And then what happens next is penetration or entry. The bacteriophage injects its nucleic acid into the bacterial cell. Okay, and that is by, for it to inject the DNA into the bacterial cell, it is helped by some phage lysozyme that will degrade a portion of the bacteriophage. And to inject, the tail portion will contract. Tail sheath contract and its core portion penetrate into the cell wall. When tail core reaches the plasma membrane, DNA moves and goes into the host. So that is happens in this entry part. And the next part is replication. The host metabolic machinery makes viral nucleic acid and proteins. And then assembly, or we can say this replication process as biosynthesis also. So initially in this biosynthesis, synthesis of DNA happens with the help of host machinery. At the same time, host DNA degrades. All the protein synthesis happens here. And the next is, as we said before, is assembly. New virus particles are assembled to form a complex virus. Head, tail, all things get attached, and into the head the phage DNA will be packed up. And the next is release. The host cell breaks open up and releases new virus particles. So with the help of phage lysosome itself, it is happening. There is another term which we want to discuss here, that is a burst period. The period from the phage attachment up to the release, that is up to 20 to 40 minutes it may take. And the number of bacteriophages is released, that is burst size. Okay, and this is the lytic cycle and the lysogenic cycle. Lysogenic cycle is also very simple. The first process is entry. That is bacteria, and bacteria having that DNA you can see here. Okay, and by the entry of this, the viral nucleic acid becomes a part of host chromosome while it's entering, and then cell division happens as usual. Although the viral nucleic acid is inactive, it replicates along with the host cell's chromosome, and the viral nucleic acid leaves the chromosome and the replication happens. The host metabolic machinery makes viral nucleic acid and proteins. So this is about the lytic and lysogenic cycle. So bacteriophage. Its structure is here. It's have a head, capsid head. In it, the DNA is seen, nucleic acid is seen, and there is a collar and the sheath, and there is a base plate attached to it. Tail fibers is this. These tail fibers help to attach to a bacterial cell. Okay, and then there is a spikes. So this is the structure of bacteriophage. So we already say that virus that infect bacteria literally mean bacteria eater or bacteriophage. It was first described by Twert in 1915 and Herrera in 1917, and widely studied in T4 phage that affect E. coli, known by coliphage. And the reproduction, we already say that attachment, penetration, uncoating, assembly, maturation, release. So adsorption, a random collision between host cell surface and virus, and it is interacted between the receptor on the host and the molecules of the surface of virion. Receptors in the host cell have specific function: viral attachment protein within the capsid or envelope, and a receptor on the plasma membrane. Viruses are host specific. It is a complex process, and a single protein of viral got intact with single receptor molecule of the host cell membrane. Some of the virus and its cellular receptors is here. So the attachment is shown here, the landing, the attachment, the tail contraction, and the penetration, that is by phage lysosome. It makes a holes and this DNA is injected into it, and the DNA injection is shown here. Okay, this is a picture which is caught in electron microscope. You can see there injecting their DNA into the cell. And penetration. Penetration is the fusion of viral envelope with the host cell and entry by endocytosis. And virus penetrate the plasma membrane and enter the host after absorption. Uncoating happens, and some virus shed their capsid proteins, some remain encapsulated, and virus genome is transported into the site where transcription or replication can begin. And then assembly. Assembly is by collection of all components needed for the virion, self-assembling process. Like pox virus assembly occur in cytoplasm and adenovirus in nucleus. Maturation is the stage at which the virus become infectious. Maturation is happening in this assembly process, right? The DNA will packed into the head, tail, the collar, the spikes, everything assembled, and a mature bacteriophage will be formed there, and in this stage the virus become infectious. Then the virion release or bursting. Virus encoded proteins are incorporated into the plasma membrane. Nucleic acid is simultaneously released. Non-enveloped virus released by the cell lysis result in the death of the host cell. Release by budding may or may not kill the host cell. The budding is shown here. DNA is packed into the procapsid. Here, the lytic cycle is shown here. Life cycle of a T4 bacteriophages. Then retrovirus. Retrovirus is a complex virus. It is an RNA virus which causes HIV, human immunodeficiency syndrome, or human immunodeficiency happening there, and this is HIV virus, human immunodeficiency virus. They cannot function as a mRNA. Instead, they convert ssRNA into double-stranded RNA. It has an envelope surrounded by RNA genome and enzyme. Reverse transcriptase is using there, and integrase is also used to take over the host machinery. So this is the structure of RNA retrovirus. So this is a short video about telling the virus and virus replication. Hope you like it.